Hello everyone. Today we'll talk about the notch signaling in human TALL. As we know that notch signaling pathway plays a pivotal role in fundamental processes of cell fate determination, thereby may contribute to the carcinogenesis. And this is true. Deregulated notch receptor expression has been reported in a growing number of human solid tumors, such as breast cancer and colon cancer, etc. Notch activation in solid tumors and support cells, such as endothelial cells, can be stimulated by other JAK or delta-type ligands, whereas in TALL, mutations in the HD domain lead to ligand-independent generation of activated notch. We call it ICM. In both settings, small molecule inhibitors of gamma secretase prevent the generation of ICM. Most activities of ICM are mediated through a nuclear transcriptional activation complex that also contains the transcription factor CSL and co-activators of the MAMO family. Proposed activities of activated notch, it means ICM, that contributes to oncogenesis are shown in green, whereas those that repress tumor development are shown in red. Solid and dashed lines denote interactions that appear to be direct and indirect, respectively. The numbers refer to references cited in the text that support the indicated interactions. The first evidence about the role of Notch1 in the pathogenesis of TALL resulted from the cloning of TAN1, a truncated and constitutively active form of Notch1, at the breakpoint of the translocation of chromosomes 7 and 9. Its presence in about 1% of TALL cases. In this translocation, the Notch1 locus in chromosome 9 is broken so that derivative chromosome 9 retains the N-terminal domains of Notch1, including the NRR region, while the transmembrane and intracellular domain of the receptor are translocated to the derivative chromosome 7, where they are apparently expressed under the control of TCR-beta regulatory sequences. Ultimately, this rearrangement resulted in constitutive activation of Notch1 signaling. Most mutations occur in the HD domain, including exon 26 and exon 27, which are present in approximately 40% of human TALLs. This mutation can destabilize the interaction between the N-terminal and C-terminal HD subunits and results in ligand-independent activation or ligand hypersensitivity. This is called HD class 1 mutations. A second mutational hotspot is located at the 3 end of the gene, which encodes the C-terminal PAST domain. PAST domain mutations are present in about 15% of TALL samples and are typically truncating and nonsense mutations, which result in deletion of recognition sequence for proteasomal degradation of ICN1 by the FBXW7 SCF complex in the nucleus. In rare cases, NOTCH1 is activated as the result of in-frame insertion in the distal part of HD domain. This is called HD class II mutations. Alternative mechanisms of NOTCH1 activation include juxtamembrane expansion NOTCH1 mutations, also called GFE alleles. This mutation consists of extracellular in-frame insertion that displays the HD domain away from the membrane. There is also the NOTCH1 H1545V mutation located in the NOTCH1 LNRC repeats that disrupts the activity of NRR and facilitates S2 processing of the HD domain. In addition to these mutations in the NOTCH1 gene, about 15% of TALLs harbor mutations in FBXW7, which typically involve K-arginine residues responsible for the recognition of phosphorylation sites in the path domain of NOTCH1. These FBXW7 mutations impair the substrate recognition function of the FBXW7 SCF complex and impair the degradation of the activated NOTCH1. In addition, the oncogenic effects of FBXW7 mutations may extend beyond the NOTCH1 signaling pathway, and this ubiquitin ligase also mediates the proteasome degradation of additional oncoproteins such as MEG, JAN, cyclin E, Aurora A, and mTau. Finally, about 20% of TAL patients harbor either dual mutations in the HD and the PAS domain of NOTCH1, or both are NOTCH1 HD allele and a FBXW7 mutation. The combined effect of these mutations results in exceedingly high level of Notch signaling as a result of Notch ligand independent activation at the membrane, plus impaired ICN1 degradation in the nucleus. As Notch mutation is found in more than 50% of TAL patients, is Notch a useful biomarker in TALL? From the table, we can say that different studies give different answers to this question. Given this, it's perhaps not surprising that associations between NOTCH1 mutation status and clinical outcomes are subtle at best. Next class will talk about Notch signaling in other malignancies.